Hey everyone, welcome back. So today is a huge day. We are officially starting a brand new module in our course, which is Next Generation Sequencing, or NGS for short. Okay, so over the last videos, we have been playing with clean, finished pasta files, but genomes don't come out of machine looking like that. So when you send a sample to a lab, the Illumina sequencing machine spits out millions of tiny fragmented DNA pieces called reads. And along with the sequence, the machine gives you a grade on how confident it is in every single letter. So this raw data is stored in a FASTQ file, right? So today we are going to learn how to read them, understand their weird grading system, and parse them using Python. So let's get started. So you already know the FASTA format. We might have created some files in the past like this one, right? So it's just a header line and a sequence. So FASTQ adds the Q for quality. So because chemical sequencing is not perfect, the machine needs a way to tell you that like I'm 99% sure that this is an A, but I'm only 50% sure that this next one is a C, right? So to do this, FASTQ files, like FASTQ file uses a strict four-line structure basically for every single read. So let's jump into VS Code and we will generate a tiny dummy FASTQ file so you can see it with your own eyes. Right, so I have a dummy FASTQ file over here which I will be creating. So we will be doing this with open. Let's call this sample read.fastq. We will open it in write mode so we can save that file as f, then f dot write dummy fastq, right? Remember, not fasta. Then we can simply print dummy fastq created. Let's run it. And there you have it, right? So dummy FASTQ file is now created. Now let's break down those four lines, exactly what we did. Okay, so line one always starts with an @ symbol. Let's directly go to that file so we can see it. So line one starts always with a @ symbol. This is the header. Okay, it contains the unique ID of the read and metadata about the sequencing machine. Line 2 is the raw DNA sequence itself, the A, T, C, and Gs that you see here, and sometimes also N if the machine completely failed to read a base. Okay, and all then line 3 is always a plus symbol. This is just a separator. Sometimes it repeats the header ID, but usually it is just a plus sign to save space. Okay, and finally the line 4, which is the Q part of the FASTA file, that looks like absolute gibberish as you can see here. So this is the quality string basically. It must have the exact same number of characters as the DNA sequence above it. Okay, as you can see here, it's exactly the same amount. So every single character in line 4 corresponds to a DNA letter in line 2. But how does it basically like how does an exclamation mark tell us that the quality of G is at something like that, right? So for that, we will this is where people will get confused. Okay, so yeah, so but it is actually a very brilliant system. The quality of DNA read is measured in Phred score. Okay, so what is it called is Phred Phred score. Okay, so a Phred score of 10 means that a 1 in 10 chance of error, like 90% accuracy. A score of 20 means 1 in 100 chance, like 99% accuracy, and a score of 30 means 99.9% accuracy. But if a sequence is 50 bases long, so writing out 30, 30, 25, 15, 40 like that, that will take up too much space and breaks the alignment, right? So scientists map these numbers to ASCII characters on your keyboard. You might have heard of these characters, ASCII characters. If you don't know, then you can look it up. Okay. So these ASCII characters on your keyboard were used for mapping these scores. So instead of writing the number 33, they just type an exclamation mark like this. Okay, exclamation mark like this, and instead of typing 65, they can type a. Okay, right, so like that. So it allows them to pack a two-digit number into a single character, and luckily we don't have to decode this by hand. Python can do it for us. So let's get started. Let's use our old friend, which is BioPython to parse this FASTQ file and convert it into convert that gibberish basically into readable Phred scores. Okay, so let's start from bio. We will import ASCII. Okay, then we will parse the FASTQ file just like we did with FASTA. Okay, so for record in sequence IO dot parse, then we will use the sample read dot FASTQ and the format is FASTQ, right? And then we will basically print what will we print ID, that will be record dot ID, right? And then we will print uh, f sequence is equal to record dot SCQ, and inside that we will use this will basically print the first 20 bases. Okay, and we can just add these dots, right? Then quality scores, right? Quality scores underscore quality scores will be equal to, this will extract the quality scores as actual numbers. Okay, so record dot uh, letter, just follow, we will discuss this after that. So record dot letter annotations, and then we will add, I think we added double dash here, yeah, so it will be single dash annotations, and then this will be uh, Phred quality. Okay, and then finally we will print uh, quality scores, quality scores first 20 similarly, and let's just add some dots over here, right? So let's see what happens when we run this. Okay, and look at that. BioPython letter annotations that you see here, that automatically decodes the ASCII string. We can now see the actual mathematical scores for our sequence. Notice how some numbers are high, like the 30s or something like that. Not exactly 30s, but like 10 and everything, right? And some are very low. So if we have a region of our DNA with scores below 20, we generally don't trust it. Okay, so as you can see here, these are basically all below 20, not even below 20, but below 10. So this is not a very trustworthy score. Okay, so it might be a sequencing error, but not a real biological mutation. So, and that is the FASTQ file. It is the foundational data format for all modern genomics. You now know that its 4-line structure, how it works, and how to decode its Phred quality scores using Python. But what happens if your sequencer gives you bad data? You cannot just analyze it, right? You have to clean it first. So in the next videos, we are going to perform quality control and trimming and other things that will be related to this video. And yeah, so as always, the code will be available on GitHub. And if you finally understand what those weird symbol in your FASTQ file means, then please hit the like and subscribe button. And I will see you in the next video.